Welcome to the Deep Dive. We're the place where we really try to plunge into complex topics and bring you the essential insights.
That's the goal.
And today, we're taking on a journey that happens inside all of us every single day. The incredible path of protein. From the moment it hits your plate, we're going to trace its, intricate breakdown, how your body absorbs those vital building blocks.
The amino acids.
Exactly, the amino acids. And then the constant, really sophisticated recycling that goes on within your cells. It's quite something.
It really is.
So our mission, our goal for this deep dive is to truly understand this biochemical adventure.
Yeah.
We want to identify the key enzymes. Think of them as molecular scissors.
Right.
And the clever transport systems that usher amino acids into your bloodstream. And importantly, what happens when this finely tuned machinery, hits a snag.
Because it sometimes does.
It does. And our guide today is a specific chapter from Marks Basic Medical Biochemistry. It just lays out this fundamental process with incredible clarity. So think of this as your shortcut to understanding a core biological process, step by step.
Making sense of the pathways, the clinical connections. Yeah.
Exactly.
And this journey, it's absolutely critical for your well-being. I mean, proteins are so much more than just building blocks for muscle, right?
Right.
They're in this constant state of flux within your cells. They're essential for growth, providing energy, underpinning almost every aspect of health. When you understand this dynamic dance, it really helps you appreciate just how intricate and vital proper nutrition is.
Yeah.
It's not just a simple one-off digestion. It's this perpetual cycle of breakdown, absorption, renewal that keeps your body functioning.
Okay, let's trace this path then. Our journey starts in, a surprisingly hostile environment.
Right.
Your stomach.
Us. Very acidic. This is where the initial assault on dietary proteins really kicks off. You imagine the proteins you eat as these long, complex chains, way too big to be absorbed directly. So to make them usable, your body needs this specialized set of tools, proteolytic enzymes or proteases, as you said. They chop these big chains into smaller pieces.
And it's fascinating how your body handles these powerful enzymes. Many of these digestive proteases, they're actually created in inactive precursor form. We call them zymogens.
Zymogens, yeah.
And this is just a brilliant protective mechanism. I mean, think about it. If these enzymes were active right away when they were made.
They digest the cell that made them.
Exactly. They'd literally start digesting the very cells that produce them. So they're secreted into your digestive tract in this safe, inactive zymogen form, and only then are they activated where and when they're needed. It's biological precision at its finest.
That is incredibly smart. And in the stomach, this activation is a really elegant chain reaction. You've got your chief cells secreting pepsinogen.
The zymogen form we just mentioned. And at the same time, your parietal cells are pumping out hydrochloric acid, HCl.
Loads of it.
Loads of it. And this acid does a couple of really crucial things. First, it denatures the proteins. It basically unfolds their complex 3D structure.
Yeah, it makes them accessible.
Exposes those internal peptide bonds, makes them easier targets for the enzymes. But here's where it gets really interesting. The HCl also acts as the trigger. It causes pepsinogen to actually cleave itself.
Right, autocatalysis.
Turning into its active form, pepsin.
Yeah.
This self-activation is what we call autocatalytic activation.
Exactly. And once pepsin is active, it goes to work as an endopeptidase.
Endo meaning inside.
Right. It doesn't just snip off the ends of the protein chains. It breaks peptide bonds within the protein, sort of chops the long loops into smaller segments. Now, it works broadly, but it does particularly favor breaking bonds near aromatic or acidic amino acids.
Okay.
And the result, those large dietary proteins are now significantly chopped down into much smaller polypeptides.
This whole initial step, it's so fundamental, and you can really see its importance when things go wrong. Like even if someone's getting enough calories, their body can still suffer profoundly if they don't get enough protein or can't use it.
And this brings us to a really stark clinical example, kwashiorkor.
Kwashiorkor.
Right. This is a severe form of protein deficiency. You often see it in children who are getting enough calories, maybe from carbohydrates like rice or cassava, but critically not enough protein.
So energy is there,
Yeah.
but the building blocks aren't.
Precisely. And the symptoms are truly heartbreaking. You see significant muscle wasting, a drastic decrease in plasma proteins, especially albumin.
And that causes swelling.
Yes, exactly. This leads to a fluid imbalance, causing excess fluid to accumulate in the tissues. That's edema. You often see this as that characteristic distended abdomen, which can make the child look kind of plump despite being severely malnourished. Without enough dietary essential amino acids, the body is literally forced to break down its own proteins muscle, plasma proteins, to get what it needs. Plus, this condition can impair the production of new digestive enzymes and healthy intestinal cells.
Oh, creating a vicious cycle.
Exactly. It makes absorption even worse.
That really drives home the point. It's not just about eating enough food,
Yeah.
but the right kind of food.
Yeah.
Okay, so leaving the acidic stomach, our journey continues into the small intestine. This is where the next crucial phase of protein digestion happens.
And here, the exocrine pancreas really steps onto the stage. It has a dual role. First, it secretes bicarbonate.
To neutralize the acid.
Right, it neutralizes that very acidic stomach content that just arrived. This is vital because it creates an optimal, slightly alkaline pH environment, which is perfect for the pancreatic enzymes to do their work.
Okay.
Second, it releases its own potent cocktail of inactive pancreatic proteases again as zymogens.
More zymogens.
More zymogens. These include trypsinogen, chymotrypsinogen, proelastase, and procarboxypeptidases. Quite a mouthful.
Wow, okay. Another elegant activation cascade, I bet. It sounds like a precisely choreographed biochemical dance.
It really is. It all starts with an enzyme called enteropeptidase. Now, this one is actually secreted by the brush border cells lining the small intestine itself.
Okay. Not from the pancreas.
Correct. And enteropeptidase specifically cleaves and activates trypsinogen into its active form, trypsin.
Okay.
And here's where trypsin earns its central role. Once activated, trypsin acts like the master switch. It cleaves all the other pancreatic zymogens.
The name of trypsinogen, proelastase.
Procarboxypeptidases into their active forms.
Yeah.
Beyond trypsin, elastase, and carboxypeptidases. It's like a domino effect, ensuring all the enzymes get ready to work pretty much simultaneously.
That's efficient.
Very. And what's remarkable is the specialization of these activated enzymes. Trypsin, chymotrypsin, and elastase are all endopeptidases, remember?
Breaking internal bonds.
Right. Continuing the work of breaking internal peptide bonds within those smaller polypeptide fragments. Trypsin, for instance, is quite specific. It prefers bonds after amino acids like lysine or arginine.
Okay.
Chymotrypsin is a bit broader, but tends to favor hydrophobic amino acids. Elastase targets smaller amino acid side chains like alanine, glycine, serine.
So they each have their preferred spots to cut.
Exactly. Then we have the exopeptidases, specifically carboxypeptidases A and B. Now, unlike the endopeptidases, these are like meticulous molecular scissors, removing amino acids one by one, but from the carboxyl end of the peptide chains.
The C-terminus.
The C-terminus, yes. Carboxypeptidase A often snips off hydrophobic amino acids, while B prefers basic ones. Collectively, they just dismantle those peptide chains from one end.
So we've got enzymes chopping in the middle, enzymes snipping off one end, a lot of specialized enzymes working together. But that's still not the final step, is it? Our own intestinal cells get involved too.
That's right. The small peptides that result from all this pancreatic enzyme action are then further broken down by enzymes produced by the intestinal epithelial cells themselves.
Okay.
On the brush border of these cells, that fuzzy lining we find aminopeptidases, they're kind of the opposite of carboxypeptidases.
So they snip from the other end, the amino end.
Exactly. They cleave amino acids from the amino end of peptides, the N-terminus. And even smaller peptides, like didases and tripeptides, can actually be absorbed directly into the intestinal cells.
Ah, interesting.
And inside the cells, intracellular peptidases finish the job, breaking them down completely. So the combined action of all these proteolytic enzymes, stomach, pancreas, intestinal brush border, inside the intestinal cells ultimately yields the individual free amino acids your body needs.
That is thorough. Now, here's a truly surprising fact, something that puts the sheer scale of this into perspective.
Yeah.
I always think about the protein I eat, but our bodies are constantly processing so much more internally, aren't they?
Absolutely. Your body is an incredible recycling machine. The amount of protein that's digested and absorbed each day isn't just from your diet. It's also from your own digestive juices. Those enzymes are proteins. And the intestinal cells that are regularly shed and broken down.
Slowed off.
Yeah, sloughed off and recycled. This internal load can be equal to or even greater than the protein you consume in your diet, easily 50 to 100 grams daily.
Wow, 50 to 100 grams recycled internally.
It's this continuous massive internal turnover that just happens without you even realizing it.
This whole digestive and absorption process, it's clearly so delicately balanced. And we can see what happens when it's disrupted in a real-world condition like cystic fibrosis.
Yes. Let's take the case study of Susan F., a young child with cystic fibrosis. This genetic disorder affects chloride channels, and in the pancreas, it causes the secretions to become incredibly thick, viscous, and dry.
Like sludge.
Pretty much.
Yeah.
This leads to an obstruction of the pancreatic ducts, and the consequence, those crucial pancreatic enzymes we just discussed, they can't reach the small intestine.
So digestion just stalls.
For fats, carbs, and proteins, yes. It causes severe malabsorption. Clinically, you'd see symptoms like foul-smelling, bulky stools because of all the undigested food failure to grow properly, and low levels of serum proteins.
But there's treatment.
Oh, yes. The good news is that supplementing with enteric-coated microspheres of pancreatic enzymes, basically tiny capsules that protect the enzymes from stomach acid and release them in the intestine, can dramatically improve her condition. It allows for proper digestion and absorption to happen.
Okay, so once all these proteins are expertly broken down into individual amino acids,
Yeah.
how do they actually get from the gut lumen across that intestinal wall and then into our bloodstream? They can't just float across, can they?
No, you're right. It's a highly regulated active process. It mainly involves two primary mechanisms: secondary active Na plus attendant transport and facilitated diffusion.
Okay, let's unpack that sodium-dependent transport first, because that sounds pretty ingenious.
It is.
So on the luminal membrane, that's the side of the intestinal cell facing the gut contents, right?
Correct, the apical side.
Okay. Specific carriers there work to co-transport both amino acids and sodium ions into the epithelial cell together.
Right, a symport system.
And what powers this? You need energy, right?
Yeah.
But it's secondary active transport.
Exactly. The power comes from a very low intracellular sodium concentration. And that low concentration is actively maintained by the NAM plus K plus ATPase pump. This pump is located on the other side of the cell, the serosal membrane, which faces the blood.
The pump is on the backside.
Right. It constantly pumps sodium out of the cell using ATP directly. This creates a steep sodium gradient, low sodium inside, high sodium outside in the gut lumen.
Yeah.
So the amino acids essentially hitch a ride down this sodium gradient using the energy stored in that gradient to move into the cell. This allows the cells to actually concentrate amino acids inside, even pulling them in against their own concentration gradients sometimes.
Clever. So the ATP energy is used indirectly to pump sodium out, which then powers amino acid uptake.
Precisely. And once inside these intestinal epithelial cells, the amino acids then need to get out the other side into the interstitial fluid and then the bloodstream. This happens primarily through various facilitated transporters, also located on that serosal membrane. These don't require sodium, but help the amino acids move down their concentration gradient out of the cell.
So it's not just one generic carrier for all amino acids, is it? It sounds like there's quite a bit of specialization and maybe even redundancy built into these transport systems.
Absolutely. Your body doesn't put all its eggs in one basket. There are at least 6 different Na plus dependent carriers identified on that apical brush border membrane.
6 different ones.
Yeah, each designed with overlapping specificity for different classes of amino acids like neutral ones, proline and hydroxyproline, acidic ones, basic ones, and also cysteine. And what's really clever is that most amino acids can actually use more than one of these systems.
A backup plan.
Sort of a built-in backup, yeah.
Yeah.
If one system isn't working perfectly, maybe another can pick up some of the slack.
And just like with the digestive enzymes, these amino acid transport systems can also have genetic defects, leading to specific conditions. Can you tell us about cystinuria?
Sure. Cystinuria, like in the case study of David K., is a genetically determined defect. It's in a specific transport protein known as cystin B0+.
B0+.
Okay. And this transporter is crucial for handling cysteine. Actually, it transports cysteine, which is 2 cysteines linked together, and also the basic amino acids, arginine, lysine, and ornithine. COAL is sometimes used as a mnemonic.
Okay.
Now, the defect impacts both the intestinal cells and, critically, the renal tubular cells in the kidney.
So it affects absorption and reabsorption.
Exactly. So these amino acids aren't efficiently absorbed from the gut, but perhaps more significantly, they aren't properly reabsorbed from the filtrate back into the blood by the kidney tubules. So you end up finding hyperaminoaciduria, high levels of these specific amino acids in the urine.
But not necessarily high levels in the blood.
Not necessarily because they're being lost in the urine, not building up in the blood, so you don't always see hyperaminoacidemia. The major clinical problem, the reason it causes symptoms, comes down to cysteine. Unlike the other 3, cysteine is much less soluble, especially in acidic urine.
So it precipitates out.
It precipitates out and forms crystals, which aggregate into painful kidney stones, or renal calculi. That's the main issue in cystinuria.
Ouch. Okay. And there's another related transport disorder mentioned affecting a different group of amino acids.
Yes, Hartnup disease. This is a relatively rare autosomal recessive disorder. It involves a defect in the transport of neutral amino acids, think alanine, valine, tryptophan, phenylalanine, across both the intestinal and renal epithelial cells. System B0 is the transporter affected here.
Neutral amino acids. And what happens clinically?
Well, interestingly, many individuals who are identified through newborn screening actually remain clinically pretty normal throughout life. But some patients can develop symptoms, often triggered by poor nutrition or illness, that resemble pellagra.
Pellagra. Isn't that niacin deficiency?
Exactly. The symptoms typically include a photosensitive rash, problems with coordination, that's ataxia, and various neuropsychiatric issues. This happens because one of the neutral amino acids affected is tryptophan.
Ah, the precursor.
Right. Tryptophan is the vital precursor for making niacin, vitamin B3, which in turn is essential for synthesizing NAD and NADT, critical coenzymes for countless metabolic reactions.
So if you can't absorb tryptophan properly.
Your body can't make enough niacin, leading to these pellagra-like deficiency symptoms. Treatment often involves niacin supplementation and making sure the person eats a high-protein diet to maximize absorption through any residual function or alternative pathways.
Fascinating link between transport and vitamin synthesis. So once these amino acids are successfully absorbed into the blood, how do they get distributed? How do they get into all the other tissues that need them, like muscle or liver?
It's largely via very similar mechanisms. Amino acids circulating in the blood are transported into other body tissues using, again, families of NAM plus dependent cotransporters and facilitated transporters embedded in the cell membranes of those tissues.
Same principles as the gut, basically.
Very similar principles, yes. These systems allow tissues like liver and muscle to actively concentrate amino acids from the blood, building up their own intracellular amino acid pools for protein synthesis or other metabolic uses. And interestingly, these transport systems aren't always just one way. They can be bidirectional. Meaning, for example, during periods of prolonged fasting or starvation, muscle tissue might break down its own protein and release amino acids back into the blood, using these transporters in reverse to supply fuel or precursors to other more vital organs.
So the traffic can go both ways depending on the body's needs.
Correct.
Are there any other, maybe less common ways things can get absorbed from the gut?
Yes, though usually it's only trace amounts. It seems small polypeptides, maybe even tiny intact proteins, can sometimes pass into the blood. This might happen either through direct transport across the intestinal epithelial cells, maybe via a process called penocytosis, like the cell drinking little bits of the gut contents,
Okay.
or possibly by slipping between the tight junctions that normally seal the gaps between the cells lining the gut wall. This is generally minimal, but it can be problematic, particularly for premature infants whose gut barrier might be less mature.
What problematic?
It can lead to allergic reactions, essentially the immune system reacting to undigested foreign proteins that manage to sneak into the bloodstream from their food.
Right. Makes sense. Okay, we've covered a huge amount about digestion and absorption, essentially getting proteins broken down and into the body. But once those amino acids are inside our cells, the story is far from over. You mentioned earlier this idea of constant flux.
Protein turnover.
Protein turnover. Your body is like a constant construction and deconstruction site. So let's shift focus now to what happens inside the cells, this continuous building up and breaking down of proteins.
That's absolutely right. Every single one of your cells maintains an intracellular amino acid pool, and this pool is constantly being replenished from two main sources: the dietary amino acids we just talked about absorbing, and crucially, from the continuous degradation of existing proteins within the cell itself. It's not static at all. All proteins have what we call a half-life, T12.
Meaning how long they last.
Essentially, yes. It's the time it takes for half of the molecules of that specific protein to be degraded and replaced. And this varies hugely. Some proteins are incredibly short-lived, with half-lives measured in just minutes, often regulatory proteins that need to change levels quickly. Others, like structural proteins, can last for days or even longer. So this constant synthesis and degradation, this turnover, is a fundamental ongoing process in every cell.
Can you give us some examples of just how significant this protein turnover is in our bodies overall? It sounds like a lot of activity.
It is immense. Consider hemoglobin in your red blood cells. Red blood cells themselves have a lifespan of about 120 days, and the hemoglobin within them is subject to damage and turnover processes. Or think about muscle proteins that are dynamically broken down to provide amino acids for energy during periods of fasting.
And then rebuilt after eating.
Exactly, swiftly resynthesized after feeding or exercise. And as we touched on earlier, think about your own digestive enzymes. Those are proteins that get secreted, used, and many are degraded. Plus, the cells lining your entire gastrointestinal tract are constantly being shed and replaced every few days.
Wow.
That represents a huge amount of internal protein turnover and recycling, often far exceeding the amount of protein you actually consume in your diet on any given day.
So why? Why does the body go to all this trouble? Why constantly break down and rebuild proteins? Why not just build a protein and have it last indefinitely, assuming it doesn't get damaged?
That's an excellent question. There are several key reasons. Firstly, this continuous turnover allows your cells to adapt swiftly to changing physiological demands or environmental signals.
Like changing metabolism.
Precisely. Cells can rapidly induce the synthesis of needed enzymes or, just as importantly, rapidly degrade enzymes that are no longer required, perhaps in response to hormones, nutrient availability, things like fasting or feeding. It allows for fine-tuning of metabolism on a relatively short timescale.
Okay, adaptability. What else?
Secondly, it provides a crucial quality control mechanism. Proteins can become damaged over time due to oxidative stress or other factors, or they might misfold during synthesis. Degradation pathways remove these damaged, misfolded, or simply unnecessary proteins, preventing their potentially toxic accumulation and ensuring cellular health. It's fundamental cellular maintenance and responsiveness.
Okay, quality control and adaptation makes sense. So how does the cell actually do it? How does it decide which proteins to degrade, and what are the main molecular machines involved in this intracellular protein breakdown?
There are really two major pathways that handle the bulk of intracellular protein degradation. First, there's lysosomal protein turnover, which often involves a process called autophagy.
Autophagy, self-eating.
Literally, yes. Lysosomes are essentially the cell's sophisticated recycling centers. They contain a whole arsenal of powerful hydrolytic enzymes, including proteases called cathepsins. In autophagy, cellular components, including old organelles or aggregates of proteins, get engulfed into vesicles.
Like little garbage bags.
Sort of, yeah. These vesicles, called autophagosomes, then fuse with the lysosomes. And inside the lysosome, the cathepsins degrade the contents, breaking proteins down into amino acids that can be reused by the cell. This pathway is particularly important for degrading long-lived proteins and organelles, and it ramps up significantly during times of stress, like starvation, allowing the cell to recycle its own components for survival.
Okay, so lysosomes handle bulk degradation and recycling, especially under stress. What's the other major pathway?
The second, and perhaps more selective pathway for many proteins, especially short-lived regulatory ones, is the ubiquitin proteasome pathway.
Ah, ubiquitin. I've heard of that. It's like a tag.
Exactly. It's a remarkable system. It involves a small, highly conserved protein called ubiquitin. Think of it as a molecular tag or label designated for destruction.
Okay.
Through a coordinated enzymatic cascade involving three types of enzymes, E1, E2, and E3, ubiquitin molecules are covalently attached to the target protein that's been selected for degradation. Often, multiple ubiquitins are added, forming a chain. This is called polyubiquitinylation.
So it gets decorated with these ubiquitin chains.
Right, and this polyubiquitin chain acts like a very clear flag or signal saying, take me to the proteasome.
So ubiquitin is literally the flag saying this one needs to go.
Yeah.
And then what is the proteasome? What happens to these flag proteins?
The proteasome is the executioner, if you will. It's a large multi-subunit protein complex shaped like a barrel or cylinder. It functions like a sophisticated molecular shredder.
A shredder, okay.
Once a polyubiquitinated protein is delivered to the proteasome, here's where energy comes in. The degradation process itself is ATP-dependent.
Needs energy.
Yes, energy in the form of ATP hydrolysis is required. It's thought to be used partly to unfold the tagged protein and then to actively thread it into the central core of the proteasome cylinder. Inside that core are active proteacytes that chop the protein into small peptides, typically about 7-9 amino acids long.
And what happens to the ubiquitin tags?
What's really elegant is that the ubiquitin molecules themselves are usually cleaved off just before degradation and released intact. They're immediately recycled to tag other proteins. It's a very efficient system.
Reuse, reduce, recycle, even at the molecular level.
Absolutely. The resulting small peptides are then further broken down into individual amino acids by other peptidases in the cytoplasm, rejoining that intracellular amino acid pool we talked about, ready to be used again for new protein synthesis or even for energy.
Is there any logic to which proteins get tagged by ubiquitin? How does the cell know?
That's a huge area of research, controlled largely by the E3 ubiquitin ligases, which provide the specificity. But one common feature found in many rapidly degraded proteins is the presence of specific amino acid sequences. For instance, sequences rich in proline P, glutamate E, serine S, and threonine T, often called PES sequences, are frequently found in proteins with short half-lives.
Pest sequences,
Yeah.
like a built-in degrade me quickly signal.
Exactly. These sequences aren't just random letters, they often act as signals recognized by the ubiquitination machinery, effectively serving as a kind of built-in expiry date, signaling to the cell that this particular protein is meant to be rapidly turned over via the proteasome.
Wow. This has been an absolutely incredible deep dive. I mean, from the acidic churning in the stomach, the intricate dance of enzymes in the small intestine, the sophisticated transporters, and then these complex recycling plants within our very cells. It's just clear that our bodies possess remarkably sophisticated, elegant, and efficient systems for managing protein.
It's truly mind-boggling when you break it down.
It really is. It's this continuous, dynamic process, far more complex than just, eating protein.
And it really highlights that this isn't just some abstract, basic biological process. It's the absolute critical underpinning of your health, your nutrition, and indeed why certain diseases manifest the way they do when these pathways go wrong. The sheer precision, the coordination, even the redundancy built into these systems are truly astounding.
Yeah.
Understanding these mechanisms really helps us grasp how vital every single step is, all the way from your plate to the cellular powerhouse.
So a final thought for you, our listener. Thinking about this sheer complexity, the efficiency, the constant turnover, what does this really tell you about the dynamic nature of your own body? And how might understanding these incredibly intricate systems maybe change the way you think about what you eat or how your body recovers and heals? Something to ponder.
Definitely food for thought.
Thank you so much for joining us on this deep dive into the world of protein metabolism.